So I will try to speak quite clearly and slowly. And if it is difficult to understand what I say, then if you can let me know at the time immediately, and I will try to say it in a different way if possible. Does that sound okay?
That sounds great, Ulrika. This is Alexandra.
Cool.
Nice to hear you. And it would be nicer to see you also, but you sound really good.
Yeah.
Alexandra is going to the... Have you heard her?
Yes, just that she sounds a little bit far away.
Okay, she will repeat it.
Hello. Can you hear me now better?
Oh, perfect.
Nice
yeah.
to see you again, Ulrika.
Likewise.
What it was the previous speech was mine about IVDR as an introductory, talking about what actually IVDR is, and I was explaining that you will talk about LDT tests and actually the validation of everything that is not considered IVDR as a reagent, and how we're gonna have to work from now on only with other these regulations, and we start, of course, panicking about it.
Yeah, I think we all feel a bit panicky. It's quite natural, I think. Yes. Yes. Okay. Shall I start?
Yes, please.
Okay. So hello, everybody. So my name then is Ulrika, and I work in the UK, in Bristol, which is here, and it looks a bit like this. We have a little harbour and a river, and by this red arrow is the hospital where I work. And as I just mentioned, I'm very happy to share what we do in our laboratory, and we try to base that on the ISO 15189 and on H62, which is the flow cytometry clinical assay guidelines from the CLSI. We do our very best, but of course, this is just our interpretation of what we do, and we're happy to work with people to find ways forward together. There we are. So, what works in our laboratory? I think all of us feel the same way. Validation is important, but very difficult to carry out at this level unless we have appropriate staffing. And that can be up and down. Some days we have sufficient staff, and other days we do not. And our way to deal with that and many other things when it comes to validation is to work out the template. So we have this quite lengthy template you can see here. Can you see my arrow on the screen as a pointer?
Yes, we do.
Yeah. Okay, good. Okay, good. Thank you. So you can see there's many pages in this validation document. Not all assays will need all fields populated. But the idea with this template is that it has taken the 15189 and the H62 guidelines and any other relevant things that we need to do for our laboratory and put that into this one template so that we know everything that needs to be done. And then, because it's very busy and we don't always have staff, sorry, there's an insect, sorry. Right, so because we don't always have staff, we work like this. For example, we have a new assay, we need to create some limit of blank experiment, right? So then we make a worksheet for each and every experiment we need to do. So we have a worksheet telling what to do, ready to go, so when we have a little bit of moment and when we have a person free, that person can just grab the worksheet and then get going. And thereby, little by little, we fill in this template. We then hope to finish the template and we can say the assay validation is complete. and we try to focus on one assay at a time. And that is how we deal with the sheer amount of work that needs to be done. And we feel it is possible. We have 2 or 3 assays already done, and we are in a good way with the next one. And so I will now show you some entries from this template. And it starts by declaring the purpose of the assay. Here is an example of the monocytosis assay validation summary. So we first need to defend why we have this assay, and that explanation or defense needs to include why can't we just buy an already CE validated assay from the shop. Okay, so why do we have to have an in-house developed assay? Well, often the answer is because there is no commercially available assay. But even in cases where there is an assay commercially available, for example, there is a plasma cell MRD assay available, I think. We still like to use our own because we have more colours, we just need one tube, so it suits our clinical needs better than the commercially available assay. So we just need to explain why we're using an in-house developed assay as opposed to a commercially available assay. And we try to put in as many references as possible, and we describe the assay a little bit. I should say now I'm very happy to share these templates after the meeting as well, if you would like to. Then we go on and we explain what the assay measures. So for example, in the monocyte assay, we say we want to measure the whole mature monocytes, and then within these monocytes, we measure relative proportion. So quite a simplistic approach, just to say what is it that we measure, and then how. And that might be a silly question, right? Because it will always be by flow cytometry. But then we specify a little bit, saying it is a label-lyse acquire processing method. And for some assays, like for example here, accessory assay, we do flow cytometry, so the immunophenotype evaluation of T cells, but we also do absolute enumeration via dual platform. So sometimes you have 2 types of measurements in one assay. We have to say also in what type of samples that we should apply the assay to. And I'm not sure about the habits in your labs, but if you do hemato-oncology, I guess, like me, we might generally use a certain type of sample, maybe bone marrow to look for MBS, or maybe blood to look for CLL or something like this. And when it comes to validation, we just need to be very crystal clear. For example, validation for this monocyte assay is carried out only on peripheral blood. So I need to be clear on that. I need to say, I'm going to use this assay on peripheral blood. If I want to, I can use it on a bone marrow sample, but we don't have full validation data for that. And I think that's okay, so long as I make that very clear. Going on, we also need to state what anticoagulants, etc. that we use. And there are many more entries like how old the sample can be and how the sample should be stored. These are quite common things, and I'm happy to share those with you after the talk as well. Another very important entry is the assay design and traceability. So traceability, now we are really in the area of ISO, right, 15189. And I think all of us in flow cytometry agree that we do not really have full traceability for flow cytometry assays. But what we can say is that we base them on clinical correlation and peer-reviewed publications. I want to speak a little bit more about that on the next slide. As for the assay design, we tend to say which antigens and reagents and the strategy for processing that we use include instrument setup, reporting, gating strategies, and so we say where we found that, or if we have invented it ourselves, what was the basis for that, what papers inspired us. So it's a little bit like a review of how we designed the assay, but shortly formulated. Of course, we talk about flow cytometers and software, etc. as well. Now, about the traceability, as I said, there is no metrological traceability, but the ISO guideline does give us suggestions for what we can use, and I'm quite happy for my assays if I can find a peer-reviewed publication from even a single laboratory that, you know, let's say has done a monocytosis assay and can show how that worked for them to separate CMML from reactive monocytosis, for example. And I can then apply clinical correlation in my own setting, meaning I can make an audit. How well did my laboratory's flow cytometry result tally with the clinical diagnosis at the end of the day? Oh, sorry about that. I'd be even happier if I can find a peer-reviewed inter-laboratory validation study, so something that's based on more than one laboratory. And if that assay was very similar to my assay, that would be great. And of course, I would be super happy almost if I could find an assay that had been validated in a laboratory network, so more than one laboratory, and it's been peer-reviewed, and the assay, I think, perfectly suits the needs for my hospital, so I can use the assay, you know, almost exactly as it is in the paper. So that would be great. And if there's then also an international consensus guideline for this, that is almost as good as it gets for us in clinical flow cytometry. So we work in the UK, at least, I'm sure you have something similar in Greece, that we have multidisciplinary team meetings where the clinicians and the diagnosticians discuss diagnosis and or what therapy to use for the patient. And with the help of the MDTs and with the outcome of the MDTs, any way you can get hold of the final diagnosis in your setting, we can carry out a clinical correlation with our flow cytometry data. As a minimum, for any assay, we'd like to show diagnostic accuracy or trueness using at least 5 negative and 5 positive cases. Okay, so when it comes to the actual assay, the reagents and the panel we're going to use, in multicolor flow cytometry, we have lots of different requirements to check that the reagents are okay. We need to titrate them to the correct volume, and then we need to see how well they fit together. And steric hindrance is something that comes up now and then. Is it really necessary to look for that? I would argue it's a very quick little test and that it sometimes can show unexpected results. And the key thing here goes back to what I mentioned before. If we set up an assay in-house that already has been published and we basically just copy and paste an already validated assay, then probably I don't need to do so many checks. So in my validation documentation, I can say not required because this is an already validated assay in an interlaboratory study. I would be happy with that. But it's quite quick to do. And if I have designed the assay myself, if it's our own in-house design panel, I always do this Vera Kimbrell's check. We also do FMO checks. It's very helpful to see where we're going to set the gates as well. Something that we perhaps were not so good at doing before that I feel we have improved on since we became ISO accredited is to have more of a sort of approach of always evaluating risks. So our validation, our validation template also include a section on risks, and they look at the laboratory health and safety. It also looks at any regulatory requirements. What is the risk category, for example? Is this low risk, medium risk, or high risk? It's possible that you discussed that already in the IVDR talk, and where the majority of flow assays in the European new directive are located. I think that's medium risk. As far as forgetting about the regulations for a moment, but thinking more about what we perceive as a high-risk assay in our laboratory, is there anything in our assay that has a high risk for incidents? For example, if we have an assay we're going to use for cerebrospinal fluids, if you just make